Okay, I'm going to teach you the steps to the scientific method. You should have this sheet in front of you, and you're going to fill in notes as I put them on the board. Okay. The first thing before you do the scientific method is you've got to have a problem. What is the problem you're trying to solve and get an answer to? Okay, so I made up a problem for you as I was looking at my fish tank over here. My problem is, I wonder how I can reduce the amount of algae in my fish tank. Okay, so I can do an experiment and see if I can get the amount of algae to be less than it is now. Okay, so the first thing we're going to do, now, first of all, for the scientific method, I put 5 steps. Depending on which book or what teacher you are listening to, there could be more or less steps. It just depends on how much do you group together. Okay. The word data encompasses a lot of things. It means collect your data and analyze your data. So I lump it together. Some put data, and then the next thing is analyze your data, come up with a conclusion. To me, that's all deal with the data, figure out what the data means. Okay, so mine, I only have 5 steps due to kind of condensing things together. Okay, number 1 on your sheet should be observation. And with the word observation, you're going to use your 5 senses. So when you're filling in your sheet, so you're going to use 5 senses to observe. And also, this observation means 2 things, not just, looking with your eyes or hearing or tasting, depending on the experiment, your 5 senses. It also means to gather information. Okay, you've got to be able to gather information. You want to see what have other people figured out, okay, in the past. And so maybe you don't have to reinvent the wheel. The experiment we were doing in class with the dancing raisins. What do you already know? What do you already know about vinegar and baking soda? What do you know about raisins? Okay, you knew they were wrinkly. You knew they would collect some bubbles. Okay, so what do you already know going into this? So I put example over here off to the side on your paper. As we go through this experiment, I want you to write down the example that I give you. Okay, so the problem, I wonder if I can reduce the amount of algae. My observation, what do I see? My observations are that I have algae in my tank. These are my observations. That, what do I know about algae? Well, it's green. Okay, I can see that. What does that tell me? It's green. It's a plant. It needs sun. Okay, what else do we know about algae? Do we know anything that actually eats algae? Okay, there's a fish, a Plecostomus. In short, it's the suckerfish. Okay, so the suckerfish eats algae. That's something else I know. Okay, so go with what you already know. Those are your observations. And then you work your way down. Knowing this, I'm going to make my hypothesis. When we did our experiment in class, we were guessing. Why do we think those raisins were moving up and down? That's your hypothesis. That's an educated guess. Okay, so as you move down your sheet, second step, hypothesis. It is an educated guess. It is not just a guess, because it's educated because you did all this background work. You gathered all the information that you knew prior to making your guess. And you can call it a guess, a prediction, an educated guess, or an educated prediction. This is what I predict is going to happen if I do this. Okay, so what do I think is going to happen if I do this? And in our example, in our experiment, we thought, well, the raisins that had the most wrinkles would dance up and down the most because they could collect the most bubbles. Okay, so that was our educated guess about it. You could also, what you also have to have in a hypothesis for it to be a good hypothesis, it must be testable. If you can't test it in an experiment, it is not a good hypothesis, okay? Next, once you, oh wait, we got to put our example down over here. You can have lots of different hypotheses. And if one doesn't work, you didn't get the results you wanted, go back, pick another one, see what happens. Okay, so what I put down, knowing what I know already about algae, I wrote down, if I put a sucker fish in my tank, I will have less algae. That's my prediction. That's my educated guess what's going to happen. We'll have to find out if I'm right or wrong. What's another hypothesis that we could write down? We know that algae is a plant that likes the sun. Okay, well, what if I make a hypothesis? The less sun on my tank, I will have less algae. Okay, test it, see if you're right. But you can only do one at a time. So I'm going to go with the sucker fish. So let's write down an example of a hypothesis. An example of my hypothesis is going to be, if I put a sucker fish in my tank, I will have less algae. Okay. Next, because it's testable, we can test this. We can put a sucker fish in our tank. Okay. We're going to try it out. That's the experimental stage. When you do an experiment, you have two groups. Okay. So the two groups, you're going to have a control group and you're going to have an experimental group. Okay, under the control group, I said on your sheet, this is the group that is not exposed to any type of change. You just leave it the way that it is. Why? Okay, I asked you why. Why do we want to have a group that we do nothing with? Answer is, you need something to compare to. If I do an experiment, how do I know if it worked if I can't compare it to something? How do I know I have less algae if I can't see where I started? Okay, so you need something to compare to. All right, so that's the control group. You do nothing with it. It has no change whatsoever done to it. Okay, next, now you're going to actually change something. This is the experimental group, and on your paper, it says you change only one variable. Okay, actually, I want to write that down. Change only one variable. You're going to change just one thing. Okay, now I ask you why. Why don't we want to change 2 things, 3 things, 4 things? Why you only change one thing at a time is so you can see what made the difference. If I put less sunlight on my tank and I put a sucker fish in there, I won't know which one caused less algae. Okay, so I can only do one thing at a time, see if it works. If it doesn't, go back, try something else. Okay, so you can only change one variable at a time. I'm going to put for my answer here. So why do we do this? If you change more than one, if you change more than one variable, you won't know what caused the change. And unfortunately, I'm running out of board. So I'm going to erase what we have so far up here. You can always rewind the video if you need to see that again. And hopefully you have that written down, or you might have to rewind. And I'm going to start up here with the word experiment. And we know that we had two groups, and we just wrote down all about, we had the control and we had the experimental group. Okay. Okay, we already wrote that down. We want to keep going. Underneath experimental group, it says, we have to fill this in on what is being changed. We know we can only change one thing at a time. That one variable that is being changed, that is called the independent variable. A lot of times we just shorten that and we call it the IV. Okay, so the independent variable is the one variable that is being changed. And on your sheet it says what is being changed is called, okay, so what's being changed is called the independent variable. Next, the response to that change. If you change something in the tank, something should happen. Okay, so that response, whatever's happening, is called the dependent variable. So the response is called the dependent variable, which we usually just put DV for short. So when you look at what is the difference between a control group and an experimental group? The experimental group has an independent and dependent variable. The control group does not, because we did not change anything. Okay, the one other thing I want you to put down on your paper, I didn't put a line for it, and I should have, I meant to. Underneath dependent variable, it is the response. It's what happens because of the change. I want you to put, it has to be measurable. So put that underneath. The dependent variable is something you can measure. So I'm going to measure the amount of my algae, okay? And I put a little dash line on your paper because I wanted to add some other information, but it doesn't go underneath just the word experimental. Okay, the next word that I want you to know that goes with doing an experiment is the word constants. Constants are variables, okay? They never change, and they're found in both groups. So both groups have constants. The experimental group has an IV and a DV, okay? So a lot of kiddos mix up the word constants and control group. Okay, so a control, you are keeping everything exactly the same in a control group. This is an actual group of the experiment. Constants are variables that don't change. And I will show you what I mean by that. The example that you're going to put off to the side, we're going to show what would our experiment look like that we're setting up. Okay, so I'm sure you can see in our experiment, we're going to have, well, I wanted them to be more looking the same than that. Okay, so we're going to set up two groups, right? A control group and an experimental group. So we'll call this our control group, and this will be our experimental group. Okay, so both groups have constants. These are variables that both of them have, and they have to stay the same. They don't change. Okay, so for example, I'm going to give both of my tanks the same amount of light. Okay, both of my tanks get rocks. Both of my tanks get a plant put into their tank. Okay, and both of my tanks have the same amount of water. Okay, it's not like one of our tanks gets a little bit of water and the other tank is totally full. All of these things that we just put down, all of those are constant variables. They don't change. And we'll list those out in a minute, just so you have them on your piece of paper. But remember, our experimental group, we have to change one thing and see what happens. So the one thing we're going to change, we said in our hypothesis, is we want to see if adding a sucker fish will give us less algae. So in this tank, we add a sucker fish. We don't up here because we want to compare how much algae does this tank have compared to this one when we put a fish in it, no fish. Okay? So the fish is the independent variable, the one thing we changed. Now the dependent variable, remember it only belongs to this group. The dependent variable is what's the response? What happened? Okay, so hopefully our response is, I'm going to put the word hopefully because we have to collect our data. Okay, the response we hope to see is less algae. Okay, so we would collect our data, see what it says. Let's just write down, if I go over here, I better go like this. Both tanks have constants, which are the same. It says same amount of water, sun, rocks, and plants. Those were all of our constants, okay? We put all the same things in, and then we just change one thing, which is our independent variable. Okay, I'm going to have to erase because we're running out of room on the board again. I'll start over here so you can still write that down. Okay. When you're doing an experiment, you're going to be gathering your data. In this instance, in this experiment, we're going to be measuring the amount of algae in our tank over an extended period of time. Okay, so after experiment, we have the word data. Okay, which for me, it's going to encompass a lot of things. It means I'm going to collect my data. I'm going to analyze my data. I'm going to figure out what is my data saying to me. Okay, and the best way to analyze your data is to put it into a graph form. Because every day I'm going to check my algae and see and measure how much it is. Okay, so I'm going to have lots of different measurements, and just reading the measurements is kind of difficult to analyze what it's saying. So 3 different types of graphs that are that we can choose from to use. The first one on your sheet is a bar graph. A bar graph is used when you're comparing data. I know that's already on your sheet, but I'm just going to write it down anyway. Bar graph is for comparing data. The next one, let me just write this. Let me just show you over here. A bar graph is where you have the bars going up And you've got your measurement off to the side. You're going to label. You're going to label down here what everything means. You're going to have a title. And what I always require and ask that you do is put the number on the top of every bar. So it's very easy to say or to see how far you really wanted it to go up. It might be between two lines. And what did you really want? Did you want that to be a 2? Did you want to be a 2.5? Did you want that to be a 2.6? Okay, so always put a number on the top of your bar. Next, a line graph. A line graph is going to be used when you're looking at change over time, over an extended period of time. Maybe you're checking something every month. You're checking something every year. Every second, you're checking something, okay? That's when there's change over time. And what a line graph looks like is maybe you're going to check your algae once a day for a week. So you would have Monday, Tuesday, Wednesday, Thursday, Friday. And how much was your algae? Okay, what we'd like to see, if our hypothesis is correct, we'd like to see that we continuously see less and less and less algae with that suckerfish. And then you would connect your lines. You'd show your measurement off to the side. You'd label what these are. Put a label on the side. Give me a title. And you'd also want to put a number on the top of every dot, just to clarify exactly what you wanted me to see. Okay, the next one is a pie chart. A pie chart is used when you want to show percentages. Okay, and when you're making, at least when you're making a pie chart for me, it doesn't have to be exact, but it needs to be proportionate, okay? We all know that that's 50%. So if we know that's 50%, and we're trying to get something to be, about 12%. If you do this and call it 12%, that's not very proportionate. That's right there in the half. So we know that this is 25% right here. So if you wanted about 12, I don't know, I'm not very good at this, just freehand, but go with what you know. This is 50%, cut it in half. This is 25%, so cut it in half again. That's giving you your different percents. So you probably need a key to show what each one of these spots is trying to represent. You can color code it and then say, okay, this black percentage right here stands for the amount of people the percentage of people wearing black shirts today versus the percentage of people wearing yellow shirts today. You'd probably want to put a key to tell me what all these different colors mean. And always have a title. Okay, on your paper, I put never forget mix and dry. Very important. Okay, let me show you mix and dry. Okay, when you make a graph, you need to know where, or when you're reading a graph, where is the independent variable? Where is the dependent variable? Okay, that's what you have to show. So, mix and dry. Here is mix and dry. When you're making a graph, you always put the independent variable on the x-axis. Okay, maybe you can't remember which is the x, which is the y-axis. Maybe an easy way to remember is this is the y-axis going up and down because it can you can make a y out of it. Okay, this is the leg. So this is the y-axis. This is the x-axis down here. What these letters stand for, to remind you, where do you put things when you're graphing to make sure you got it on the right spot? The M stands for well, actually, it's probably easier to start with the I. The I stands for the independent variable. The independent variable always goes on the x-axis. Okay, that's what the x stands for. And the independent variable is what is being manipulated. It's the one you're changing, right? You're changing one variable at a time. So the M stands for manipulated. So the independent variable goes here. Very easy then. The Y is for the y-axis. Okay, so the y-axis is where you put the dependent variable, and it is the response. If I change this, this will happen. And remember, it has to be measurable. So when you've got your graph, this is where you're putting your numbers on the side. Okay. So the one thing I am changing goes down here, and then my numbers would go off to the side. Okay. These are going to be, maybe I would be measuring it depending on the experiment. Maybe it's measured in milliliters. Maybe it's measured as a number. But that's what your label is going to tell us. Your label would tell us if it's in inches versus centimeters versus milliliters. And this is the measurement over here because that's the response. And then the very last thing on your paper, we had data written down. And then the last thing is to communicate your results. Because with your data, we've already came up with a conclusion by reading a graph, and we're going to communicate our results. We definitely want everybody to know what we did so they don't have to, reinvent the wheel and do it all over again. Or maybe they would like to repeat our experiment and want to know exactly what we did. So you communicate. If you were a scientist doing an experiment, you would maybe get published, okay? Publish it in a journal so other people can read it. Make a presentation, make a speech somewhere and present your data and your conclusions to others. But you've got to get the word out, hey, I did this experiment. And if it's not, remember, if your conclusion is not what you thought, my hypothesis was that the suckerfish was going to reduce my algae. And then based on my data, the amount of algae actually went up. Okay. And you're like, oh, well, that didn't work. Okay. Go back, make another hypothesis. Maybe it is the amount of sun. Okay. Go through the whole thing again and only change one variable at a time. Okay. I hope you got all that down.